Alright gang, this is section 16.2. I'm actually going to split section 16.2 into 3 different parts. So this is part A. Hopefully it doesn't take too long here. This is Sophie beside me. Can you look right there and say hi?
Hi.
Yeah, she woke up pretty early this morning, and so she is joining me for this video. And you're going to be still, right?
Yeah.
Okay. Anyway, as we left it before, Watson and Crick had made their double helix model of DNA, and they published a paper about it, got a Nobel Prize, and they made a prediction. As soon as they came up with their double helix model, they made a prediction, and their prediction was that if they looked at the structure of DNA as they had it with the A's and the T's and the C's and G's matching up, I need you to be quiet. Be quiet, okay? Alright. Anyway, they predicted that when it came to the replicating of DNA, so if we're going to make a new cell, we need to make a new copy of DNA, they predicted that the DNA would just, we'd break the hydrogen bonds that were holding the A's, T's, and C's and G's together. So break those bonds, which would happen here, and then you could bring in new nucleotides that matched up with the original nucleotides. And a term there, if you have something that matches up, like the A's and the T's match up to each other, those are called complementary. So we would say complementary nucleotides come in and match with the original on each strand. And so in the end, when you have your new DNA, or you have your 2 copies of DNA, you have 1 copy of DNA here that is part parent strand and part new DNA, and then the other side is also part parent DNA and part new DNA. So those, they call that model the semiconservative model, because each new strand or each new DNA molecule is part old, part new, or it's part conserved and part new. So that was their prediction, although they had no evidence of that other than just the structure of DNA made sense for it to work that way. So there was actually some other people came up with some different models, different ideas of how DNA would work to, how DNA would work to make replication happen, and the next slide is going to get into that. Alright, so this is a diagram showing the other models of DNA replication. We have the one in the middle here that, where are we at here? Yeah, the semiconservative model that I already explained, the idea that the original DNA would split into separate sides, okay? And then if we're looking at this first generation of DNA that would result, it would be part old and part new, okay? And now I can't see the screen. Hold on, Seth. Anyway, and then if you went to a second generation or a second division, you would actually have these splitting, and you'd have part of the original parent strand in one of these, and part old, so you can see the semiconservative, and you'd have completely new DNA in the second generation here, or the second cellular generation. Okay, we're not talking about organism generations, but anyway, so that's a semiconservative model. Other models, the one was the conservative model up here, and that I like to call the photocopy model, where you've got the original, you make a photocopy of it so the original is intact, and then if you go to the second generation or second round of replication, you would have just a complete copying of each of them. So that's the conservative model. And then they have another model, I don't know who came up with this one because it doesn't make any sense to me, but the dispersive model that basically says you start out with your original DNA, and as it makes new DNA, it's just kind of each strand of new DNA is just part old, part new, and it's just kind of random. And if you go into the second generation, so this would be half old, half new, but in kind of a random way. If you go to the second generation, this would be a quarter of the original and 3/4 new, a quarter of the original, 3/4. If you went out another generation, it would be 1/8 the original DNA and 7/8 new, and so on, and it would just kind of get dispersed, if you will, through the generations. So those are the different models. There had to become a way of testing this, and so we're going to go through the Meselson stall experiment. And if you read through the textbook, you probably honestly just kind of skipped this because it was really just a diagram. But this is one that, again, understanding scientific procedure and inquiry, and what they did was a pretty genius experiment, and so it is valuable for you guys to hear it and hopefully to understand it, so we'll go through that next. Okay, so the Meselson-Stahl experiment, again, they're trying to figure out conservative versus semi-conservative versus dispersive model of DNA, and basically they had to have a way to distinguish between the original parent DNA versus when you got daughter cells, what's the new DNA that's going into them. So what they came up with as an idea was to use different weighted nitrogen, and so they grew up bacteria. And they grew it in a media here, so there's bacteria in a media that included an isotope of nitrogen that was heavy nitrogen. So it was nitrogen 15, it had an extra neutron in each of the atoms, and that made it heavier than nitrogen 14 that you're seeing here, and so they could distinguish how heavy the DNA was in these different cells based on the nitrogen that they took up. Okay. So what they did is they grew bacteria in nitrogen 15 until they knew that all of the bacteria was completely nitrogen 15 containing. So its DNA was heavy, had heavy DNA. And then what they did is they transferred it into a vial that had, or a flask actually, that had light nitrogen in it. And they know that the E. coli bacteria that they used had a 20-minute life cycle basically. So it would, in 20 minutes, it would go through replication and it would have new DNA. Okay, so after 20 minutes, they isolated the E. coli from the nitrogen 14, and they put it in a centrifuge to see how heavy it was. Remember, the heavier something is, the further down or up in the centrifuge tube that it's going to settle. And they found that it settled, in our diagram here, that it settled at about the halfway point on this test tube, or the centrifuge tube. Then they also collected more bacteria out of this vial after 40 minutes. And what they found there was then there was two different weights of DNA. One weight was continued to be at the medium level, and then one weight that was really, really light because it was up at the top. So they had to interpret this. So our next slide is going to interpret that, these findings and what it actually supported. Alright, so now we're going to look at how what they found fits in with the different models, and you can kind of see the conclusion here with the X's through things. But let's go down to the dispersive model first of all. Okay, actually no, we'll do the conservative model first of all. If we look at the conservative model, what we would have expected after one generation of growth is we would expect that there would have been heavy bacteria, okay, that contained just the heavy nitrogen, and there would have been completely light bacteria, because in the conservative model, if you remember, the original DNA stays completely together and we just make a new photocopy. So the new DNA would have been light, the old DNA would have been heavy. So right away after the first 20 minutes when they did the centrifuge tube, they would have known that, okay, it can't be the conservative model, and so they crossed that one out. So that was that. If we look at go down to the dispersive model. If we look at the dispersive model, after one round of replication, you would have DNA that's not heavy, which would have been down here, and it's not light, up here. We would have had DNA that was half old, half new, so it would have been medium, and that's what they found. So after 20 minutes, they thought, hey, dispersive model, it could actually be the dispersive model, something that's a medium weight. Okay, but then what you would expect after the second round of replication is you would expect a DNA that is 1/4 original DNA and then 3/4 heavy DNA. So you would have had something, you would have expected that all of your DNA would have been at a like 3/4 light. And anyway, so it would have been not medium and not super light, but it would have been at about a 3/4 level. Anyway, because this is how the DNA would have looked, and this is not how the experiment worked out. They didn't get one band that was like a 3/4 light and 1/4 heavy situation, so they knew it wasn't that. So then if we match this up with the semi-conservative model, they would expect that the original DNA would have been, there's my original DNA, would have been split and half of it going with each of the new strands, so you should get a medium band of DNA when in the centrifuge tube, and they did get that, a medium band, indicating that it's half heavy, half light in each of the DNA molecules. So then what they'd expect in the next round is that when these go through the next round of replication, the old parent DNA is going to be present in half of the DNA molecules, or 2 in this case, of the DNA molecules, and the other half are going to be completely light. And so you'd expect 2 bands, one that's medium, these would be the medium ones, and then one band that would be of the completely light. DNA. And that is what they observed, and so this was our evidence that the semi-conservative model is true, and that the DNA actually unzips and complementary nucleotides come in and replace. And if I bring this back up here, put it over top, that this is what's happening. Unzip, okay, complementary nucleotides come in and match up with the original strands, and then we end up with two strands that are half old and half new. So that's it for this video. The next video is going to be looking more at the details of how this process happens, because it gets pretty complex, and we will kind of leave it at that. If you're still listening, I'll give you a quick tip that I put some animations in the animation section of the website, and they are about replication, and they say chapter 16 on them, and they're going to go very well either for you to look at before you watch the next video or right after you watch the next video. So that's that. Sophie, can you say goodbye?
Goodbye.
Can you look right there? Say goodbye.
Goodbye.
Goodbye. All right. See you in class.